Hello everybody, we're going to go over test 4 from spring 2014. Have the answers already highlighted on here. The first 30 questions are over Unit 10, so for all of these, this is referencing for the first 30 Unit 10, and then the last 10 are from Unit 11. So the first one, which does not participate in the digestion of dietary lipids. These first 4 here are found on slide 9, which is talking about the digestion of lipids, so make sure you know all of those reactions and their enzymes and products really well. Lipoprotein lipase is something I didn't see anywhere in the unit, so that's the answer. It's the odd man out. This molecule is synthesized only in the liver under normal conditions. A lot of these are pancreatic or can be synthesized in most cells. Glycocholate is a bile salt. Other thing to know about that is that they are cholesterol derived. You'll see the full reactions for glycocholate in Unit 11, how you make those. Which lipoprotein particles are responsible for reverse cholesterol transport? So make sure you know the functions of all of these. HDL, the so-called good cholesterol, does reverse cholesterol transport. Maybe a couple answers from slide 13 there. Which protein is phosphorylated by pKa? So a lot of these, this is asking from the mobilization of fatty acids slide. So perilipin or hormone-sensitive lipase are the two that are directly phosphorylated. You might throw in ATGL, and that's actually activated by perilipin directly, so that wouldn't be an answer. This particle had the highest proportion of liver-made lipids. This question comes up a lot, that liver-made lipids part, because chylomicrons are dietary lipids, not made from the liver, so that's not the answer. VLDLs is the answer. LDL is highest portion cholesterol, HDL, highest portion of protein. This is just to confuse you. This reaction is part of the reassembly of dietary lipids. So without knowing anything here, there's only one. I mean, you have the carnitine shuttle that shows up twice, something from the citrate shuttle. None of this have to do with dietary lipids, and we haven't seen any reactions with VLDL in the actual reaction. So you can process of eliminate. You know it's D. D is also the only one going from a mono triglyceride to a diglyceride. Mono to di. That's the reassembly process. Those reactions are seen on slide 10, but I wouldn't worry about memorizing them. Just know the principle. You're building single fatty acids up to triglycerides again. Which cells do not have the capacity for fatty acid oxidation? Brain cells. Brain cells are the only ones. They're going to run off only glucose or ketone bodies. Couldn't find that anywhere in the slides, but he says it a bunch of times, and he'll ask about it twice here in the test. Which enzymatic process does not take place during the beta oxidation of palmitate? So activation and transferring or transporting are going to be for both beta oxidation and the synthesis reactions. So those aren't going to be answers. And then remember, OHOT is the beta oxidation ones. Reduction is not part of that. Reduction is part of synthesis only. The process of fatty acid oxidation is not mitochondrial. That's activation. So they're activated in the cytosol, carried into the mitochondria by the carnitine shuttle, and then all of these are beta oxidation reactions that all happen in the mitochondria. Protein is normally bound to the inner mitochondrial membrane. You see a picture of that on slide 27. It's the translocase involved with the carnitine shuttle enzymes. This vitamin is needed for the activation and the beta oxidation of palmitate. So these are needed for just beta oxidation, niacin and riboflavin. Biotin is one of those. It's for the odd number fatty acids. It's used for the carboxylase. And pantothenate is anything involving CoA, which is the reaction, and CoA is in the beta oxidation as well. So that's going to be the answer. Metabolite is not a fuel for brain cells. Oleate, you could put any fatty acid here. So glucose and ketone bodies here are going to be used as fuel, and then acetyl-CoA and pyruvate can be used either through the electron transport chain, or you can reverse it through gluconeogenesis. Either way, they can be fuel. Palmitate, stearate, those are going to be the other popular ones you'll see here. Which metabolite's concentration is lower than normal in the blood during starvation? So it's glucose. Glucose drops during starvation. The ketone bodies are significantly increased, and palmitate is barely increased. This molecule does not have a mitochondrial carrier. It's kind of a comprehensive question. We've seen malate gluconeogenesis. We know the ketone bodies do, because they're taken from the liver cells, mitochondria, to the cytosol in the blood and back into the mitochondria. So those both have carriers. And we see pyruvate in the citrate shuttle. And we saw it in glycolysis as well. So acetyl-CoA does not have its own carrier. This liver enzyme is found inside the mitochondria and the cytosol. Thiolase is the one that you see in the synthesizing a ketone and using a ketone as fuel. So that's the answer there. See it on these two slides. Metabolite does not appear to be a fuel molecule. So while acetone is a ketone body, it's not a fuel. The hydroxybutyrate and acetoacetate are the two ketones that we use for fuel. The acetone's kind of like a ketone byproduct that you get rid of by breathing out. Enzymatic step is not catalyzed by fatty acid synthase. You can either know everything by fatty acid synthase does, or any of the beta oxidation reactions would be the answer. Which molecule is the most immediate precursor for the synthesis of palmitate and cholesterol? So we know palmitate and cholesterol are both synthesized in the mitochondria. So it's weird how we gave both a mitochondrial and a cytosolic version of it. If you see on slide 86, it shows the locations of where everything happens. So make sure to know that slide really well. Most likely we'll be pulling a question from it. Protein missing in the liver is the CoA transferase. See on slide 57, that's the reason why the liver can't use the ketone bodies it creates. Which protein is not a part of the citrate shuttle? I highly recommend memorizing the whole citrate shuttle and all the peculiars about some of these enzymes, and knowing that acetyl-CoA carboxylase is in the fatty acid synthesis activation, not in the shuttle. Enzyme does not appear to be regulated. HMG-CoA synthase. The main regulation you need to memorize is on 97 and 98. That's the summary of it. Any of these slides that I put in this column for these regulation questions are kind of the illustration of it, or where exactly it is. One that you don't see in Unit 10 is HMG-CoA reductase. Yeah, it's a Unit 11 one involved in the cholesterol synthesis. Which metabolite is not part of the citrate shuttle? Another question, so make sure you have that memorized. Melanyl-CoA. Which metabolite is synthesized by all cells? Hopefully you could answer this one on the first test, because this is used in glycolysis. Glycolysis is happening in all cells. Enzyme is known to be inhibited by phosphorylation. You'll see the particulars to that on slide 101. ACC, also on 97 and 98. Which lipid process takes place mostly in the cytosol. It's the fatty acid synthesis. These two, A and D, are both in the mitochondria, and desaturation is in the ER. I think elongation takes place in the ER and the mitochondria. How many net molecules of ATP are created by the complete oxidation of stearate? So to do this, you need to 18 minus 2 divided by 2. So 16 divided by 2 is 8 rounds. So that means there's going to be 7 rounds plus 1 last round. For each of these, you can see the details of this on the ATP slide for where exactly all this is coming from. For all the other rounds, it's 14 ATPs are generated. So 14 times 7 is 98. And then in the very last round, there's going to be 24 ATP created. So we're going to do 98 plus 24 is 122. So that's where a lot of people will put D as the answer. But for all of these, you need to do minus 2 for the 2 ATP used in activation. So this says net molecules. So whatever number you get here, no matter what, minus 2 and 120 is the answer. Which metabolite is known to partially inhibit ACC? Palmitoyl-CoA. See that in all the regulation at the end. Serine enzyme is cyclooxygenase. You will find a way to ask about this in some way. Maybe what inhibits it? Aspirin. This molecule is not needed to convert cytosolic acetyl-CoA to palmitate. You can know the ACC uses biotin, and this bicarbonate here, ATP is required, malic enzyme creates NADPH. So you can know all of that, or you can know that in fatty acid synthesis there's a dehydration, because in beta oxidation there's a hydration, and they're inverse. So that's the way I would know it, but that is kind of a tricky question. Which lipid enzyme is known to have a requirement for B12? That's the mutase. So make sure you know your odd chain fatty acid requirements and your unsaturated or polyunsaturated fatty acid requirements. For the unsaturated, you need 2 enzymes, the reductase and the isomerase. And for here, you need the mutase, isomerase, and the carboxylase. Carboxylase requires biotin. 10 quick questions on Unit 11. Most immediate precursor of triglycerides and glycerophospholipids. So what you're going to see on slide 3 is that DHAP is the most immediate precursor. That's not in the options here. So all of these guys are pretty far away. They happen in the second half of glycolysis. This is pretty early on, and this is right before DHAP. So that's the answer. That's a tricky one, but he will make sure you know the pathways on Unit 3, and I have that outlined pretty well in the video for how I memorized it. Glycocolate, all of these things are required, but no vitamins. Very last slide of the unit talks about the isopentenyl pyrophosphate, and vitamin C is not on that list, but he always pulls a question from that last slide. Which reaction is essential for the synthesis of triglycerides and neutral glycerolipids? You're going to see that on slide 3 as well. Which protein is missing from liver cells? You could have asked this one on the first test as well. GLUT4 is muscle and fat. GLUT2 is liver. So you didn't even need to read the rest of the other potential answers. Which metabolite is not an intermediate in the synthesis of cholesterol? Malonyl-CoA. That's only for the fatty acid synthesis. All of these are in the cholesterol pathway. Degradation of gangliosides is the only clinical thing. It's on slide 21, so he's going to ask about it in some way. Usually he asks to make sure you know it's a ganglioside and not a globoside. Which vitamin is not used in the metabolism of lipids? Make sure you know where all of these are used. We haven't seen folate yet. I think that's reserved for the last or second to last unit. Signaling molecule cannot be synthesized from membrane lipids. Membrane lipids being cholesterol. Remember, epinephrine is tyrosine derived. All of these other ones are lipid derived. Some of them that he asks about most commonly are on slide 34. And the last one, which of these molecules does not appear to affect HMG-CoA reductase? So this is the key enzyme in the cholesterol synthesis pathway. So make sure you know that really well in slide 28. You can know it just by aspirin. Aspirin is the odd man out here. If he throws in a weird one, it's most likely going to be a statin, and statins do inhibit that enzyme. So just watch out for that. There's the negative feedback with liver cholesterol. There's the AMP kinase that we saw in Unit 3, and insulin, epinephrine, and glucagon affect pretty much every regulation, so that's not the answer either. Good luck on the test. See you next time.